Passive transport. Passive transport can occur directly across the membrane or with the help of a transport protein. It occurs without energy expenditure by the cell because substances are moving from high to low concentration. A limited number of substances can diffuse right across the phospholipid bilayer through simple diffusion without help from a transport protein, for example, oxygen and carbon dioxide. When the oxygen concentration is higher on the outside of the cell than the inside of the cell, for example, when a cell is metabolically active, oxygen diffuses rapidly into the cell. Conversely, carbon dioxide builds up within the cell as a waste product of cellular respiration and diffuses out of the cell. In humans, blood transports both of these substances. When we breathe in, oxygen enters into our bloodstream, and when we breathe out, carbon dioxide is expelled. Lipid-soluble substances such as hydrocarbons may also be able to diffuse through the membrane. For substances that cannot freely cross the bilayer, which is most substances, a transport protein is required. If a substance crosses the cell membrane by moving down its concentration gradient with the aid of a transport protein, it's called facilitated diffusion. As mentioned previously, transport proteins include channel proteins such as ion channels and aquaporins, and carrier proteins such as glucose transporters. Note that facilitated diffusion is a passive process. Even though a transport protein is involved, there is no energy expenditure required because substances naturally move down their concentration gradient. Water is so important in living systems that its movement has its own special term. The diffusion of water across a selectively permeable membrane is specifically referred to as osmosis. Now, I know it sounds odd that water can diffuse down a concentration gradient given that the environment itself is made of water. In the case of water, however, the presence of dissolved substances affects the concentration of free water molecules. This is because the water molecules will form hydration shells around the substances. This is what causes them to dissolve, thereby decreasing the number of free water molecules. Thus, water naturally moves from an area of low solute concentration with a high number of free water molecules to an area of higher solute concentration with a lower number of free water molecules. Osmosis can be observed with the use of a U-tube, different from what you might think of when you think of YouTube. In this example, there's a semi-permeable membrane down the middle of the U-tube. The membrane is permeable to water, but not to sugar. Initially, one side of the tube has a higher concentration of sugar, so the water is drawn to the side with a greater solute concentration. Water will diffuse across the membrane until the right side is diluted enough that the sugar concentrations, and hence the free water molecule concentrations, are equal. Once equilibrium has been achieved, water molecules will continue to move back and forth across the membrane, but there will be no net movement of the water. The ability of a solution to cause a cell to gain or lose water is referred to as tonicity. A solution that causes a cell to gain water is hypotonic. A solution that causes a cell to lose water is hypertonic. And a solution that has no effect on the passage of water into or out of the cell is isotonic. Note that tonicity is relative. If a solution is hypotonic, then the cell is hypertonic relative to the solution, and vice versa. Tonicity is important in a number of living systems. For example, red blood cells are floating in a substance called plasma. It is important that they be isotonic relative to the plasma. If the plasma is hypotonic, water will move into the red blood cells, causing them to burst. If the plasma is hypertonic, water will move out of the cells, causing them to shrivel. Tonicity is also important in many organisms. Paramecium, for example, is a single-celled organism that lives in an aquatic environment. When it enters into a hypotonic environment, water will rush into the cell, and the paramecium must pump water out using a contractile vacuole so it won't burst. Freshwater fish face the same problem. They're constantly gaining water through osmosis since their cells are hypertonic relative to the hypotonic water. They counteract this water uptake by drinking very little and peeing a lot. In contrast, saltwater fishes lose water to their environment. They make up for this by drinking a lot and peeing very little. They eliminate the excess salt through their gills and their kidneys. Recall that in a hypertonic solution, there are fewer free water molecules in the solution than there are in the cell. Therefore, water will move out of the cell where there are a greater number of free water molecules, down its concentration gradient to an area where there are fewer free water molecules. Now let's try a little harder question. Consider the following scenario. The solutions in the two arms of this U-tube are separated by a membrane that is permeable to water and glucose, but not to sucrose. Initially, the liquid levels on both sides are equal. Initially, both sides of the U-tube are isotonic. Although there are different concentrations of each sugar, the overall concentrations are the same, and therefore the number of free water molecules is the same on both sides. However, because the membrane is permeable to glucose, the glucose will diffuse across the membrane until there's an equal concentration of glucose on each side of the U-tube. Sucrose, however, which is a disaccharide, cannot pass through the membrane. Therefore, the water will move to the side of the higher sucrose concentration and continue to move until the sucrose concentrations are equal on both sides. Thus, both A and C are correct. If you have any questions about any of this content, please don't hesitate to contact me.